So here are the answers to the questions from the previous chapter. So why don't you pause the video, tick your answers off, and then go on to the next chapter, which is proteins. Proteins are long chains of amino acids which are folded to produce a specific shape. Proteins perform many important functions. They can be structural proteins. In other words, they can go to make things like our hair, our nails, our muscles. They can be hormones, they can be antibodies, and they can be enzymes. An enzyme is a biological catalyst, and all enzymes are proteins. The protein is folded to produce an active site which has a specific shape, and each enzyme has its own substrate. Now this is an important word. The substrate is the molecule which the enzyme works on. It may break it down, it may stick things together, but it's the molecule that the enzyme works on. And the substrate fits into the active site where the reaction takes place, and then the products are released. Now, if an enzyme is exposed to a high temperature, this can change the shape of the active site, meaning that the substrate no longer fits and the enzyme no longer works. This is called denaturing. The wrong pH will also denature enzymes. They each have their own optimum pH. Now, just be careful here. A quick word about language. It's enzymes that are denatured, not cells or plants or organs or tissues. So the enzyme is in blue, the substrate is in red. You can see that the substrate fits into the active site where the reaction takes place, and then the products are released. We also make use of enzymes in the home. Biological detergents, for example, use proteases. This is the term for protein digesting enzymes. And they also use lipases. This is fat digesting enzymes. Now, because they contain enzymes, they're more effective at lower temperatures than non-biological detergents. So you can turn the temperature down on your washing machine, so this saves energy. There are various uses of enzymes in industry. For example, baby food makers use proteases to pre-digest protein, so this makes the food softer and easier for babies to digest. Carbohydrases are used. They work on carbohydrates, they work on starch, and they convert starch into sugar syrup for use in confectionery and cakes, that sort of thing. And makers of slimming foods use an enzyme called isomerase. Now this converts glucose into fructose. Now fructose is sweeter than glucose, which means that we can put less of the sugar into the food. So therefore the food contains fewer calories. Don't say it makes the food healthier. It may not necessarily make it healthier, but it will contain fewer calories. Now, all these enzymes which are produced to use in detergents and baby foods and all the rest of it, they're actually produced by microorganisms which are genetically engineered to produce these enzymes. And if you remember from Unit 1, we talked about genetic engineering back then. Now, enzymes can be immobilized, which means they can be fixed in place. We can embed enzyme molecules on membranes or in beads, and then the reactants are made to flow over the enzyme molecules, so the enzymes are not mixed in with the reactants. And this means that the product is not contaminated with the enzyme because it hasn't been mixed in with it, and it allows the enzymes to easily be reused because they don't have to be separated again. So this shows an apparatus in which starch syrup is poured through a column of beads which have amylase enzyme immobilized in the beads. So the starch just flows over the beads, and as it does so it comes into contact with the amylase molecules, and so the starch is converted into glucose, and that means that glucose solution is what dribbles out at the bottom. Enzymes in digestion. So this is just a reminder of the structure of the digestive system. Now you might be called upon in the exam to label any of these organs. Now these are three groups of enzymes which you need to know and understand: amylase, protease and lipase. You need to be able to state which organs produce them and what the action is. So that means what's the substrate and what are the products of that reaction. Two other chemicals which are released in the digestive system are hydrochloric acid, which is released in the stomach, and that provides the best conditions for the enzymes which are released in the stomach. They like acidic conditions. And bile. Now this is produced by the liver and it's stored in the gallbladder, which is a little pocket within the liver. It's released into the small intestine. Now bile is slightly alkaline, which means that it neutralizes the acid that comes with the stomach contents into the small intestine. So this provides the optimum conditions for the enzymes in the small intestine. It also has the effect of emulsifying fats, which means breaks fats into small droplets. This increases the surface area for the enzyme lipase to digest the fats. So it doesn't digest them, but it does break them down into smaller droplets. Okay, time to test yourself then on proteins.